Hello, everyone. My name is Lucas Mullen, and I will be presenting the work Building a Genome Archiving and Communication System Integrated into a Health Information System. This work is being carried out by the bioinformatics team of the Health Informatics Department of the Italian Hospital of Buenos Aires in Argentina. Personalized precision medicine seeks to use genomic approaches to improve treatments, prevent disease, and promotes people's health. This implies the integration of different types of data with electronic medical records, which in turn opens up new lines of research and opportunities to improve health management. The development of next-generation sequencing technology and its rapid price drop in recent years allowed healthcare institutions to incorporate sequencing equipment into their laboratories and use genomic information to assist in the making of clinical decisions. At the same time, results processing and interpretation require highly complex computing platforms. Files with genetic data are used and generated both in the next-generation sequencing process and in its bioinformatic processing. These files must be managed efficiently for reasons of order, control, quality, and information security. The size of these files increases exponentially as the scope of genome sequencing increases. Thus, there is a trend towards storing complete sequencing data separate from the electronic health record. In this study, we show how at the Italian Hospital of Buenos Aires, a highly complex hospital accredited with level 7 by the Healthcare Information and Management Systems Society, the genomic data of the patients were integrated into a genomic information archiving and communication system known as GACS. So the main objectives that we set for in this work were to implement an archive unit to store the raw genomic files generated by the hospital's DNA sequencers, to implement a data storage unit corresponding to the genetic variants obtained in the sequencing processes carried out on hospital patients, and also to implement the necessary services for both uploading and downloading of raw genomic data and genetic variants of patients. In the system that was worked, the GACS store files and genetic variants in a similar way to how a picture archiving and communication system known as PACS does with the images generated by different medical services. So conceptually, a GACS is an independent system that allows the storage and sharing of raw or processed genetic information with the rest of the information systems, both inside and outside the hospital. Regarding the architecture used, the GACS is made up of two non-relational MongoDB databases and a file system. MongoDB databases store the variants resulting from genetic studies in such a way that they can be quickly consulted by a genetic information communication service. The first database stores the information from germline genetic tests, while the second stores somatic line tests. The file system is used in the GACS for the storage of raw files generated in the sequencing process and intermediate files generated by the bioinformatic processing. The genomic files that are stored in the file system are obtained directly from the DNA sequencers of the DNA sequencing laboratory of the Italian hospital. These files are, first, the BAM files, which files contain information of each sequence obtained by the sequencer and is mapped to a human reference genome. Then there is the raw UBAM files, which files are a variant of the BAM file format in which the data read does not contain any mapping information. And lastly, there is the variant call format known as BCF, which are plain text files with a specific format used in bioinformatics to store sequence variations. The details of the genetic variants of each patient are stored in two MongoDB databases so they can be quickly consulted by the services that communicate the information with the electronic medical record and the decision support system. The separation into two databases occurs because in the germline, the database is centered on the patient, and in the somatic one, it is centered on a sample from a biopsy, which in turn is related to a patient and a type of tumor. The design of the databases and the service to obtain genetic information from them was designed and developed jointly by the bioinformatics team of the Italian Hospital of Buenos Aires and by the company Genome IT. Regarding the integration process with the laboratory, a relational data model that uses Oracle database stores the metadata that relates the patient to the laboratory samples. In this model, these samples are also associated with their different genetic studies carried out and with the path of the genomic files within the GACS file system. On the other hand, a web service is used by the Laboratory Information System, known as LIS, to write in the relational model tables each time a new genetic study report is made. The main data stored in the model are sample and patient identifiers, genetic test code, test date, reporting system, sequencer equipment identifier, and name, type, and directory of the genomic files generated in the sequencing process. So in order to obtain the files from the sequencer and the genetic variants from the laboratory's reporting system, a daily computer process searches the tables for new records. Each time a new sample is found, a workflow is triggered that is responsible for searching for the reported variants and the genomic files generated in the sequencing equipment to upload them to the GACS. Finally, the metadata in the model is completed with the directories where they were saved. The following are the results obtained so far in this work. During the months of January to March of the year 2021, two next-generation sequencing ion torrent sequencers and the laboratory information system were integrated into the GACS. So considering genomic files and databases, 2.7 terabytes of data have been integrated into the GACS so far. 352 genomic files have also been associated with 63 patients. These files include BAM, BCF, and UBAM files. Among this volume of information are results of tests from the years 2016 to 2021. The information that was stored in the GACS was produced by genetic tests performed in the Italian Hospital of Buenos Aires sequencing laboratory. These tests correspond to the following gene panels: BRCA1, BRCA2 germline genetic test, cystic fibrosis test, hereditary hemorrhagic telangiectasia test, and Oncomine focus assay solid tumor somatic test. So what we can conclude by the work carried out is that implementing GACS can provide efficient and centralized storage of all genomic information of patients that ensures their integrity and their subsequent use. While it can be technically challenging and requires a lot of storage, it can aid in diagnostics, information security, research purposes, and as supporting documentation for patient studies. And that is all. For any query, you can contact us via email at the following addresses. And thank you very much.